So these are our disclosures. And now I think that we can go into the topic itself. Duchenne muscular dystrophy is a very severe condition. It's a muscle deteriorating disease, and it leads to rapid loss of mobility and loss of ambulation today in every kid where we make the diagnosis at different ages, actually, from 6 to 16, 17 years old. But unfortunately, today, all kids lose ambulation. And the estimated global prevalence is about 7 cases per 100,000 male. And there are different studies in different countries and different regions, but we can keep in mind that about 1 out of 5,000 newborn, male newborn, is affected by Duchenne muscular dystrophy. And the root cause of Duchenne muscular dystrophy is actually a mutation, most of time an out-of-frame mutation in the dystrophin gene. And because of the dystrophin gene, the cell is lacking a shock absorber, but a shock absorber with several functions, and it will be much more fragile, and it will destabilize the muscle fibers during movement. So we need to understand that the dystrophin gene is one of the biggest in humans, right? There are 79 parts that are read, and we call it exons, right? Because in a gene, you have parts that are read, we call them exon, and between them, there are introns. Dystrophin is simply a giant, 79 exons, and between these exons, the introns will be removed during a process that we call the splicing. So it's a rod-shaped protein, and the very first function of dystrophin is to provide a strong mechanical link between the muscle cytoskeleton and the extracellular matrix. So it binds with both parts, but on top of that, there are several binding sites on the dystrophin for interacting with key proteins in the muscle cells. Duchenne muscular dystrophy is not only a disease of muscles, it's also a disease of other organs at different time in life. And one probably of the most important is the brain. Cognitive and behavioral problems have been associated with Duchenne muscular dystrophy. They are different isoforms of dystrophin. Some of them are expressed in the brain and some not. So there are mutations into the dystrophin genes that are more prone to be to generate brain involvement. And we can keep in mind that the more distal is the mutation, the higher is the risk to have a central nervous system involvement. And this starts with mutations after exon 45. We can keep in mind that many kids with Duchenne muscular dystrophy, including those with mutations before exon 45, will have some kind of attention deficit, but it can go up to very severe cognitive issues and sometimes autistic features in patients with the most distal mutations. So we need to keep in mind that several types of mutations may happen in the dystrophin, and Duchenne results most of the time from out of frame. It means that you have no dystrophin that is produced. But these mutations can be deletions, which is a gap in the gene, duplication, which is the repeat of a structure, insertions or deletions of one or several pairs of base. You can also have a single pair of base that changes and that generates a codon that means stops. In general, patients with Duchenne express no or very few dystrophin. And then if this mutation does not affect the reading frame and that you can still express a little bit of dystrophin, you have a milder form that we call Becker muscular dystrophy. And you know, we've written milder with coma because actually it is clear that for some patients Becker is not mild at all. Becker patients in general express a lower amount of truncated dystrophin. So these are different types of mutations. As you can see, we have zoomed on a very specific region, which is the region between exon 45 and 50. You can miss an exon, like exon 50. And if you miss exon 50, you have definitely an out-of-frame deletion, and you will have no protein. But you can also have a deletion that is slightly larger, like for instance, a deletion of 49 to of 50 and 51. And if you're missing 50, 51, there is no disruption of the reading frame. And the exon 49 and exon 52 can be, there is no disruption of the reading frame between these two exons. So you will get a mutation that respects the reading frame. You will have a truncated dystrophin, probably in a lower amount, and this is a typical mutation of a Becker muscular dystrophy. You may have also a duplication, and look, this is a duplication of exon 50, and as you can easily understand, this disrupts the reading frame. And then you can have more subtle mutations. So for instance, if a single pair of base is inserted or deleted, then you will disrupt the entire reading frame, and sooner or later a stop codon will happen. And then you may also have a very simple mutations that affect a single pair of base, and because of that, a mutation, a stop codon will happen, and there will be no reading of the RNA. Finally, you may also have some mutations that happens in the intron, and because of the intronic mutations, having abnormal splicing of this intron and the flanking exon during the splicing process. So because of the absence of dystrophin, there is a destabilization of the membrane, an increased calcium permeability, and several downstream mechanisms. And unfortunately, this will lead to the muscle cell death. Of course, this muscle cell will be replaced by the satellite cells that are around, but these satellite cells will have the same genetic mutations and will have the same fate. So there are some mutations in Duchenne that are associated with a slightly milder phenotype, but altogether, the genotype-phenotype correlation in Duchenne is very weak. We have patients who are eligible for exon skipping 44, so which means the deletion that are flanking these exons may be slightly milder, but this is just exception, right? And in general, we say that out-of-frame mutations leads to Duchenne, in-frame mutation to Becker, but there are exceptions. For instance, if the deletion is so large that actually there is nothing remaining of dystrophin, or if it affects very specific domain. And again, maybe the most strongest genotype to phenotype correlation is the one that is associated with central nervous system issues in the most distant part of the dystrophin. And last and probably least, there are also patients with a very large mutations that not only encompass the dystrophin genes, but also the neighboring genes. Most of them are associated with intellectual disability.